Right, when we last left off, we looked at the basic structure of DNA and reminded ourselves that what its components are, and that really all of our chromosomes with their genes are inherently coiled up DNA. But we're left with a little problem, and that we really need to know, because remember from mitosis that when cells do divide, before they divide, they have to copy the DNA. Copy that entire set of instructions so that one cell can keep a copy, another cell can have the copy as well. So the question remains, how can we do that? How can we ensure that in this set of chromosomes here, a set of DNA that goes to this cell, will be exactly the same as the set that goes to this cell? And we're in luck here, because it's actually laughably simple and really, really easy. Building upon what we recalled from our activities in building a basic DNA molecule with base pairing rules, we can figure this out. So let's take a look at it. So let's remember with our base pairing rules that we had said that, remember, DNA is essentially simply almost like a ladder, with the rungs of our ladder being composed of our bases, A's, T's, C's, and G's. And remember, again, with base pairing, adenine always pairs with thymine, cytosine always pairs with guanine. So just like in our DNA molecule building activity, as long as we know one side of our base pairs, like in this case guanine, cytosine, thymine, adenine, we automatically know what's on this side. Because if there's a guanine here, there's got to be a cytosine there. A cytosine right here, got to be a guanine there. Adenine and thymine. Simple. This is so easy. So with that in mind, that's going to enable us to look at how DNA replicates itself. Very, very simply. As we just said, each side of the DNA acts as a template for the other side. So what we're going to see is it actually copies itself in a semi-conservative manner. Semi meaning half, conservative meaning save. And what we'll actually see, because the sides are opposites of one another, if we take our original strand, all we're going to have to do is open it up or unzip it and bring in new opposite complementary bases. So just as we had just seen in our images here, again, looking at this guy, let's say that there was an adenine here. All we'll have to do is bring in a brand new thymine and attach that here. And that is what is occurring here. And if we do that with both sides, if you look, we now we'll end up with an old strand of original DNA, and we'll be able to build new complementary strands. And the best part, if that old one was an A, we brought in a T. That means the other old side was a thymine. We had to bring in an A. And what you'll notice is that our strands are going to end up being absolutely complementary to one another. Perfect. Two exact identical copies. So let's take a quick look at how simple and easy this is. All we've got to do is unzip it, bring in new bases, and zip them shut. So how are we going to do that? So to replicate it, again, all we have to do is, it's really a three-step process. The first thing that's going to happen is a special enzyme called helicase will have to unzip the DNA and open it up. Then yet another enzyme, remember proteins and enzymes are amazingly important, DNA polymerase. We know what it's going to be doing, dealing with DNA. Polymer we saw in the beginning of the year, that means many or several. And ACE is the ending. So this is going to build a polymer of DNA. It's going to go down each side and it adds opposite complementary bases, and then we just zip itself back up again. So a snapshot in this we can see here is that one thing that is missing in this image that we will see is that our original strands of DNA, here actually is our helicase enzyme. It's unzipping this guy, working up that way. And notice that here's our old original strand of DNA here. There's an old original strand of DNA there. And all of these new nucleotides are coming in and pairing up. There's a G right here. Well, here comes a C. There's a T right there. There comes an adenine, an A. The only thing that's missing from this image is the fact that we need to have an enzyme right here and an enzyme right here. Those are going to be our friends, DNA polymerase. That's our important enzyme that's going to get the job done here for us and bring in new bases. But how simple is that? Unzip it, attach bases, and then all we got to do is zip it shut. It's a beautiful thing when you think about it. So with that in mind, now that we know the process, what I'd like to do is actually, forgive me here, but steal a video for my video. And please pay attention to the author here as we look at this. And we're not going to watch the whole video, but I want to point out a few things to you so you can see replication real time, if you will, in action. So let's take a look at this. So there's our DNA strand, which is working according to plan. Now when we had this, remember that first enzyme, helicase, its job is going to be to unzip the DNA. So there's our wrapped up, coiled up DNA, and as you watch, what will happen is that DNA will actually come in and will unzip the molecule. It's opened up now. It's now two distinct strands for that. So there's our DNA coiled up with all the nice bases, making that helix. Remember, our code is going to be in there somewhere. So we have our original strand of DNA. Now remember the two players here. Helicase, very, very important enzyme. That its job is going to be our wrapped up DNA here is going to be unzipped. It's going to loosen. Remember those hydrogen bonds holding it together. Uncoils it and zips it open. So that we actually now have two single strands of old original DNA, which is kind of nice. Magical. Special thanks to, thank you. Now remember, our nucleotides are floating around. Adenine, guanine, cytosine, and thymine. Here's our sugars and our phosphates. Remember our three parts. We used them to build them yesterday. What we're going to do here is another enzyme, DNA polymerase, is going to grab onto them, and notice that they're coming in here. Here comes our gigantic, enormous blob. This is our DNA polymerase, and if you look at it, look at what it's doing. Almost like a stapler, it's locking those brand new nucleotides in place. There's one going this way, so now we're going from a single strand to a double strand.